Hello, everyone. I'm Mary Ellen with Headwaters Science Institute, and welcome to today's Lunch with a Scientist. Today, we have Dr. Ameeta Parekh from the FDA, who will provide an overview of how drugs are generally developed and approved by the FDA. But before we meet Ameeta, Headwaters would really like to thank Boreal Woodward for their support of our online learning programs. They've recently jumped on board and we're thrilled to have them. They're an amazing action sports facility and ski and snowboard resort located right near our home base in Soda Springs. They do a lot of community partnerships and they work with young youth all year long. So we're really thankful to have them on board. So now to introduce Dr. Ameeta Parekh, she has a PhD in pharmaceutical sciences from the University of Maryland. She's a senior advisor for scientific collaborations in the Office of Translational Sciences in the Center for Drug Evaluation and Research at the FDA. Dr. Parekh advises the center on its scientific collaborations with internal and external stakeholders. She has served on the agency for 34 years in various scientific and leadership roles. While at the FDA, Dr. Parekh has worked in the Office of the Commissioner, where she spearheaded the effort focusing on women's participation in clinical research and in the drug development and approval area as a regulatory reviewer and a team leader in clinical pharmacology. Over the years, Dr. Parekh has contributed to several therapeutic areas and led the development of several guidance documents and policies, and she's published extensively on important FDA regulatory science issues. So we are thrilled to have her here today. And good morning, well, afternoon, Dr. Parekh. I know you're on the East Coast. How is your day going?
Oh, the day is going well. It's kind of getting a little chilly here now. The summer's on its way out, and so we are getting ready for fall and winter on the East
Oh,
Coast.
oh, lovely. Well, at least you don't have the smoke like we've got out here, which is great.
Oh, yes, great.
Yeah. So are you ready to tell us all about the FDA?
Certainly. If you're ready, should I just hit
Yeah.
slideshow or
Yep, here you go.
here
Yeah.
you go? And I can do, tell me if, how's this? You probably don't hear me. Mary Ellen, could you hear me?
Yes, you're ready to go. I'll back
Okay.
out so you won't be able to hear me.
All right. So I'm going to hit slideshow then. So hello, everyone. I'm Ameeta Parekh. I work with the Food and Drug Administration, FDA, as it's commonly known. And FDA, the Food and Drug Administration, it's one of the oldest, actually, it is the oldest consumer protection agency in the federal government. And when I say that, I don't want to scare you. I'm not going to be talking through the long history of the FDA. It has an extensive history, and I would encourage you, whenever you have the time and the interest, to go on the website. And there's lots of information about the FDA on our website. What I would like to do, though, I would like to walk you through the different centers at a very high level, the FDA organizational chart, how we are structured. I work within the FDA in a specific center. So there are many centers in the FDA. One of them is the Center for Drug Evaluation and Research, CDER, Center for Drug Evaluation and Research, CDER, as we call it. So I'm in CDER right now. I have been through different places within the FDA. I've been in the Office of Commissioner, but right now, as a CDER person, I've worked in CDER the longest, while I've been at the FDA, and I would like to give you an overview of how drugs are developed and approved at the FDA. So, my title, FDA and Drug Development. So, an outline of my talk. As I mentioned, I'm going to talk to you about FDA, what its purpose is, what our mission is, what are our responsibilities, why do we exist. So, I'll give you, based on our responsibilities, we have an organizational structure. So, I'll go through that structure, focus on the different centers, but I will do a deeper dive on the drug development and approval, which is the responsibility of one specific center, the CDER, Center for Drug Evaluation and Research at FDA. What is our mission? Why do we exist? What is our responsibility? And spend a minute here, just walk with me through this mission, the language here. So, FDA is responsible. And if you just look at the first paragraph here, it's very interesting. It covers so many aspects of consumer protection, public health. So, FDA is responsible for protecting the public health by assuring the safety and efficacy and security of human drugs, veterinary drugs, biological products, medical devices, food supply, cosmetics, and products that emit radiation. So, if you look at just this aspect, it's not just human drugs as in what you and I think of, it's veterinary drugs and biological products, devices, foods. uh, And in addition, there's another aspect, and I'm going to come to that, which is in the second part of this second paragraph. It says right here, we also have the responsibility for regulating the manufacturing, marketing, and distribution of tobacco products to protect the public health and to reduce tobacco use by minors. So, when you look at this whole gamut, you can see how broad and wide our mission is. And that's partly the reason we are on the news so much, because we have such a broad responsibility. In addition, if you see here at the beginning of the second bullet here, we are also responsible for speeding innovation and making medicines more effective, safe, more affordable, and communicating all this information, all this accurate science-based information to the public, so they can be people can be responsible using these products that we regulate. We also, and just go down to the last part of the bullet, we also have the responsibility for fostering development of medical products to respond to deliberate and naturally emerging public health threats. So, as you can see, just step back, and if you look at this mission statement, we have a very, very broad mission. And because of this broad mission, we have a very broad organizational structure. So if you look at the top here, this is the Office of Commissioner that provides the high level policy across all the centers. You don't need to really focus in and try and read each one. You can go to our website and try and understand what each of these centers do. But at a high level, here is, for example, the Center for Biologics and Center for Devices and Center for Drugs and Foods and Tobacco. And I talked about all of this earlier in our mission. And in addition to all of these centers that are responsible for the various components of that mission, we also have several of these other offices that exist under the Office of Commissioner. But most of the scientific review of applications and the scientific assessment and the determinations and the decisions are made right here in the middle by all these centers in FDA. Let me focus on a few of these centers that kind of are more defined in the mission. You'll go back and remember, hopefully, what I just mentioned in the mission statement. Center for Food Safety and Applied Nutrition, CIFSAN. So we have acronyms for all of these centers. So Center for Food Safety Applied Nutrition, CIFSAN, is responsible for foods. Center for Drug Evaluation and Research, CDER, this is where I am, is responsible for drugs. Center for Biologics Evaluation and Research, CBER, is responsible for biologics such as blood and vaccines. Center for Devices, CDRH, Center for Devices and Radiological Health, as the name suggests, it oversees devices. And then I also mentioned veterinary medicine in our mission. So Center for Veterinary Medicine, CVM, is responsible for veterinary drugs and animal feed. Let me go here to the last one and then I'll go back to the NCTR. Center for Tobacco Products is responsible for regulating tobacco products, CTP. What does NCTR do? So NCTR is the National Center for Toxicological Research. This is the only center in the whole of the FDA that's responsible primarily to do research. What is this research? Oftentimes, during the review and doing their task, the routine task that, for example, CIFSAN or CEDAR or CBER or CDRH or CVM or CTP, as they are doing their assessments, some questions come up. And those questions may require some kind of research that needs to be done. So, NCTR is the center that's primarily responsible for supporting this research, and it supports all of these FDA product centers. So that's the responsibility of NCTR, the National Center for Toxicological Research. But what am I going to talk about? I'm not going to go into details of everything. That's too much information. Besides, I work in the a Center for Drug Evaluation and Research. I'm going to talk about CEDAR. What do we do? And what does CEDAR do? And as you go back to the mission statement, CEDAR is responsible for the safety to get safe and effective drug products to public. So that's the broad responsibility of CEDAR. What do what do all these drugs cover? What are these drugs that are regulated by CEDAR? They are new drugs, prescription drugs. There's prescription drugs and non-prescription drugs. So the prescription drugs are those that the doctor needs to write a prescription for. And then there are drugs that you don't need prescriptions for, and those are called over the counter drugs. So, what CEDAR does is it has oversight for prescription drugs, new drugs, generic drugs, biologics, biosimilar products, and over the counter drugs as well. In addition to regulating all of these products, we also have the oversight for post marketing safety and promotion. So, what is post marketing? So, when pharmaceutical companies develop drugs, they have all the scientific information that they've collected. They just can't put it on the market. They have to send all the information to the FDA. FDA reviews it and CEDAR reviews it. And we say, yes, it can be approved. But beyond that, post marketing, once the drug is marketed, FDA doesn't stop at that point. FDA continues to do some post-marketing safety assessment, and that goes throughout the uh, life cycle of the drug product. And FDA CEDAR also oversees the drug quality throughout the life cycle of the product. So that's basically the broad responsibility of CEDAR. I've been talking about drug. There's a way we define this word drug at the FDA. Any product that's intended for use in the diagnosis, cure, mitigation, treatment, and prevention of disease. So that's basically how we define a drug. And this is intended to affect the structure, any function of the body. So with this definition, drugs includes prescription, over the counter drugs, and it includes generic drugs. I want to take you to a place now which hopefully you've all had a chance to think about sometime or another. For example, if you've had a drug and you've had to either take it yourself or for your relative, for your younger sibling or older sibling, or for your parents or grandma or uncle, aunt, whatever. When you have that situation, you are taking this drug or you're giving this drug to someone, there's a reason that why this drug is being administered, right? There's an indication. Is it for pain relief, or is it for migraine, or is it for diabetes? Or so there's an indication and the usage. How do you take this drug? So there's item number one there, which is the drug. All drugs have indication and the usage. How much do you take? That's the dosage, right? Do you take this much, or do you take twice that much, or half that much? So that's the dosage. How much do you take? And how do you take it? That's the administration part. Is it a tablet? So, dosage form and strength. So, what is a dosage form? A dosage form means what's the form that the drug comes in? Does it come as a tablet? Does it come as a capsule? Is it a solution or a suspension? And what are the strengths? Is it a 200 milligram? Is it a 50 milligram? So, that's the strength. Are there any concerns when I take this drug? So that is under 4 and 5, for instance. Are there any contraindications? As an example, there could be a big contraindication that could say, do not take this drug X with drug Y. And there's always some description in the labeling. If you go deeper on the right side, it describes why those two drugs should not be taken together. So that's the contraindication. And then there's the warning section and the precaution section, because it's always good to know what are some of the adverse reactions that can happen, or what I should do and be watchful of when I take the medications. Adverse reactions, for instance, when I suddenly get a rash, or if I get feel lightheaded or something like that, that's an adverse reaction to that drug. If I go into the label, I'll think, oh yeah, that's an adverse reaction, let me not just drive right now because that's the side effect of the drug. So it's good to have adverse reactions listed, and that's always there along with the information for the drug. Drug interactions. If you take 2 or 3 or multiple drugs together, what's the potential for them to interact? That's always good to know. Another thing, this is item 8 here, use in special populations. What do I mean by that? How about pediatrics? How about little kids? Or how about uh, very elderly people who could be taking the drug? Or how about people who, for instance, have kidney failure? Can they take this drug? So that falls under the special populations. And the information is always useful regarding how to use the drugs in special populations, those specific populations. Um, Overdose. What happens with overdosing? And then there's the scientific section here, and this is primarily uh, description, detailed description of the drug, such as what's the chemical structure, stability of the drug, clinical pharmacology, the pharmacology part of the drug, and the toxicology part of the drug. Note the two terms here, non clinical and clinical. Clinical means humans. So what are the studies? Then item number 3 describes the studies done in humans, and non-clinical here describes or discusses the studies done in non-clinical settings, which is in animals or in vitro tests. And then, of course, this information on how the drug is supplied, how to store it, how to handle it, and then patient counseling information. Why am I sharing all this information with you? And the reason is this is the information that is typically included in the drug product labels or the package inserts that you often see when you pick up a prescription drug from a drugstore or a pharmacy. And it's all of these little pieces that are the responsibility of CDER, Center for Drug Evaluation and Research. And CDER has a multidisciplinary group of scientists. There are chemists, and there are pharmacologists, and there are toxicologists, and there are statisticians and MDs. So, all of these people are responsible for the different sections, reviewing the different sections in the label. And once they review all of this, which is actually submitted by the pharmaceutical companies, they review it, edit it, and this is the information that's generally in the drug product labels or the package inserts. How do we make these decisions though? And there's one big bottom line thing to keep in mind. You kind of already guessed drug development is a complex, lengthy, and a multidisciplinary process. And it goes through a lot of non clinical, clinical testing. But the evidence that's generated and collected is sent to the FDA CDER to review. I mentioned we have scientists in CDER who review these things. How do we make the final decision, the ultimate approval decision, whether the drug is to be approved or if there are additional studies needed and there's still unanswered questions that are critical, or whether the drug should not be approved. So, even that sometimes is a decision we have to make. And all of these decisions, the ultimate approval decision is basically made after an assessment of whether the drug's benefit outweigh its risk. So a patient has disease, a patient is likely to take this drug for that disease or the condition. Is the benefit of that drug going to outweigh the risk of taking the drug or the risk of having the disease? So that's basically the benefit risk ratio and the the principle behind these scales here. So benefit risk ratio is what drives our decision. I'll dive a little deeper, and just bear with me because I'm repeating it enough times, and hopefully by the end of it you'll get it, and you probably have already gotten it. So look at this trajectory from this end to this end, from left to right. Look at these two vertical bars here, this bar and this bar here. This bar represents IND, investigational new drug, and this bar represents NDA BLA, new drug application or biologic license application. Let me explain to you what these segments before the red bar, between the red bar, and after the red bar mean. I mentioned a lot about the chemistry. The chemical synthesis is a part of it, getting the molecule, understanding the molecule, describing the molecule. That's what a drug is, right? So there has to be, how do you decide which drug to take forward? So there is a lot of discovery and chemical synthesis that happens. And this is what the chemists are responsible for. So they come up with these chemistry structures and they characterize that structure and they think of purification and synthesis and understanding the value of that chemical for a specific indication. What do you do with that structure or a chemical then? Well, we have to see, and all of this on the left side of this bar is preclinical. What does preclinical mean? I mentioned earlier, clinical is humans, preclinical is anything that's either in animals or in vitro or outside of human. It could be human cells in a test tube or a petri dish. So all of that kind of studies, all of those kinds of studies get done before the clinical phase, before it gets into the market, or sorry, before it gets into humans. So before it gets into humans, there's this preclinical phase where there's a lot of chemical synthesis and analysis of the molecule. There's a lot of research lab and animal work that happens here. Once all of this is done, the pharmaceutical company that's developing this drug comes to the FDA with an investigational new drug, an IND, and says to the FDA, here is the information that we have for this molecule. Here is our IND application. Can you review it and let us know if it's okay based on this information to start studies in humans? Till the FDA looks at it and says okay, and FDA has a time frame for that, and FDA has to get back to that pharmaceutical company and say, yes, you can go ahead and start human studies, or no, you cannot, because this aspect is still a concern for us, and you need to go back and address that. And then, and only then, you can start human studies. Once an IND is granted, accepted, and clinical studies get started, these are the studies that happen in humans. So, clinical development phase is all in humans, but it happens in different phases, and that's described here. So, phase one is one phase where very few humans, a very small group of humans, are given that drug. And what's the primary reason for that very few normal, healthy volunteers who get that drug? You're basically trying to see if that drug is safe. So you could be giving very low doses, and those doses could be determined from these earlier preclinical studies. So, starting dose, what can we start with? And let's assess the safety and tolerability of that compound or that chemical that was developed and sent to us by this company. So that's what is assessed in the early phases. Who is doing all these studies? The pharmaceutical companies are. FDA does not conduct all of this work. It's the pharmaceutical companies who are doing these studies. So, clinical phase one is basically small groups of healthy volunteers to assess safety, tolerability, and pharmacological studies, basically to understand how is the body treating, how is the body handling the drug, basically. How is it being broken down, or where is it going? Is it in the urine, or is it not in the urine, or where is it? So, those kinds of studies are early phase one studies. Once you get some reassurance from these early studies, then you have much more confidence in taking that drug forward. And then you get into the phase two, which is patients, but a small group of patients. So, what you're doing here, you continue to assess the safety and tolerability in the phase two, but because these are patients, you also try and gather some information about its effectiveness, its efficacy. So, efficacy is basically its intended use, the indication for which this drug is being developed. So, in this phase two, efficacy can be tested, early efficacy, some markers of efficacy, and at that point, you can also, or the pharmaceutical company can also assess what is the dose. If it's too low of a dose, you may not get the efficacy that you are intending to get. If it's too high of a dose, you may get the efficacy, but you're also getting the side effects or the toxicity. So, that dose determination is an assessment that can be done in this phase. Once there's all of that information, safety, tolerability, efficacy testing, dose determination, then the big phase 3 trials are launched. And it's these big phase 3 trials that are done in large numbers of patients, patients who will be taking that drug for that indication. And those are large pivotal trials where full blown safety and full blown efficacy is studied. And based on all of this information that gets submitted to the FDA, at this point, FDA then, the reviewers then make a determination after they review all this whether the benefits of the drug outweigh the risk. And that's when they make the determination of approval or non approval and labeling. And this stage, this bar here, is called the new drug application or the new or biological application. And so this is the clinical phase where human tests are done, a determination is made, and then once it's marketed, FDA continues to do post market evaluation of the drug. Let's get back to a little more detail on drug discovery. There's no standard route. So if you recall, this is the discovery part and the non-clinical part. Remember, this is the clinical part. So I'm going to get into these phases now. Drug discovery, there's no standard route through which drugs are identified or synthesized. It could be lab synthesis or it could be made from living materials. But this is again the responsibility for the chemist. What else do the chemists do? They follow, they synthesize the drug, but that's not the end. A drug candidate needs to be purified and characterized because you can't just keep changing the molecule. So you have to have it pure, you have to have it characterized. And that's not the end. It's a powder or a crystalline form or whatever form it's in. It needs to be formulated. What do I mean by formulated? Formulated means it needs to be in a form that can be taken by the patients, tablet, for instance, or capsule, or a solution, or a suspension, or injection, or whatever. So that formulation is a whole science in and of itself. And this could involve, all of these could involve several complex steps. It depends on the type of the drug, the molecule, and the dosage form, tablet, capsule, or any complex. There are some very complex dosage forms that can take a lot of technology and understanding and a lot of scientific work through. So this is a step that follows, just the discovery. Given an example here, but basically what I'm saying here is there are many steps and there could be very complex steps as well. Non-clinical development, I talked about it in the earlier slide. So what is the non-clinical? Remember, clinical is humans, non-clinical is before that. So that is in vivo animal studies, and it could be in vitro studies. So these in vivo animal studies is basically characterizing toxic effects. So it, we have some guidelines. Companies follow those guidelines, typically two or more species, and varying lengths of time that the drug or that chemical needs to be administered. It could be acute, which is very short term, one dose. It could be subchronic, which is like, let's give it for a week or whatever, and then chronic, but chronic being long term. But again, it depends on the drug and what kind of information you're trying to derive from these studies. Identifying and characterizing adverse effects. That's very important because that is the data that is needed in that IND, investigational new drug, the red bar, the first red bar that I talked about. Identifying and characterizing adverse events, determining the safe starting dose. What dose do we start humans? We've never given this drug to humans. Where do we start? So there's a lot of experience that's amassed over the years. And what is the safe starting dose? And even that safe starting dose, whoever the humans are monitored very closely and carefully because you never know what the reaction could be. Another aspect that's captured in these studies is called the pharmacokinetics. It's acronym, we like to use acronyms. ADME studies. What is ADME? Absorption. How is the drug? So let's say the drug is taken orally. Does it get absorbed? Or it doesn't even get absorbed? How is it distributed in the body? Is it primarily distributed in certain regions versus the other regions? Metabolism. What's metabolism? How is it broken down? So, metabolism is basically how is the body breaking down that drug? And once it is broken down, how is it excreted? How does the body get rid of that drug? Is it through urine, for instance? So, that is ADME: absorption, distribution, metabolism, excretion. And then, if we have some animal model of efficacy, proof of concept is basically efficacy. The concept is some indication that we are envisioning for that drug. Is there any proof of concept from these non clinical development? So, if it's possible to assess the proof of concept efficacy, if there is such a model, all of that can get done in the non-clinical development. However, there is a legal requirement. You can't just stop here and put the drug on the market or give it to humans. There needs to be demonstration of efficacy, and there are legal requirements for efficacy. Basically, there's law that requires establishment of efficacy of a drug as a prerequisite for marketing. And how do you assess this effectiveness? It is established by this term called substantial evidence. There needs to be substantial evidence for the drug's efficacy before it can be put on the market. What do we mean by substantial evidence? And let me define that to you. And it's legally defined. It the substantial evidence means it's the evidence that's consisting of adequate, well controlled investigations, including clinical investigations by experts qualified by scientific training and experience to evaluate the effectiveness of the drug involved, on the basis of which it could be fairly and responsibly concluded by such experts that the drug will have the effect it's supposed to have, or purports, or is represented to have under the conditions of use prescribed, recommended, or suggested in the labeling or the proposed labeling thereof. So, this, as you can see, is a very legal language. I'd given you an example of the labeling. There is an indication, there's subjects, the populations who will be using the drug. So all of that has to be based on something, and that's based on all of the non-clinical studies as well as the substantial evidence that's provided for that drug. And as I mentioned, I'll just quickly skim through the slide. There are different disciplines. It's a multidisciplinary effort. The chemistry part, I've talked to you about the chemistry part, the structure of the molecule, the synthesis of the molecule. I talked about the preclinical toxicology, where you assess the safety issues, the toxicity of the drug in from in vitro tests or in animal tests. And then there is the clinical pharmacology, the pharmacokinetics, ADME, that I talked about. And then there is this medical, which is basically the substantial evidence of how it works in humans. So all of these things are assessed by a multidisciplinary team. And then there are the statisticians. How do you make a decision that the drug works? So it has to be compared to something, and that something could be a placebo, which is a sugar tablet, a fake drug or fake tablet, or it could be an active drug which has already shown to be effective. So when those two things are compared, this is your test drug, and this is your reference product. So this is better than this, or this is as good as this. Whatever that mathematical assessment is, that is basically done by the statisticians. And then, based on all of this, the decision is made whether it's safe to proceed with approving, non approving, whatever that decision is. Who monitors all of this stuff? I mean, you could be thinking that, everyone is going off in their silos, but no, we don't. We work as a team, and there's a project manager that keeps an eye on all of the items that are going on, all of the reviews. And this project manager basically is working with the pharmaceutical company and coordinating the progress of the drug and what is needed, and keeping the communications open. And sometimes there could be other disciplines depending on the type of drug. I want to share with you something that I hope you've thought about, and if not, just think about this. So, the easiest, I shouldn't say the easiest, I would say the maximum number of compounds could be, a large number in this pre-discovery or discovery phase. Could be a large number of compounds here that the chemist comes up with. And the chemist could say, hey, I have, these many compounds, let's see which one. And they have this library of models or relationships where the structure of the molecule can be related to the effect. So they call it the structure activity relationships. So based on that kind of scientific assessment, they have all these compounds, and they say, okay, I have so many compounds in my library. Let's take it forward. Where's the first step? The first step is, of course, they do a lot of chemistry where they do dissolution, maybe solubility. Does it dissolve? Does it not dissolve? All of that stuff. Is it even stable or not stable? They go into animal testing or preclinical testing, and they may decide, hey, this is too toxic. So a lot of compounds are eliminated. So if you look at this dark blue cone, just focus on this dark blue cone for a second. All these compounds from chemistry, here is the preclinical in animals. There's only a few that make it through for various reasons. Could be toxicity, could be many other things. And then as you move forward into humans, remember the clinical trials, that's in the yellow box. Preclinical is in this blue box. This is the clinical phase. I talked to you about phase 1, phase 2, phase 3. As you can notice, the cone is getting narrower. What's happening here? More and more drugs are being eliminated for various reasons. It could be safety, it could be toxicity, it could be lack of efficacy, or it's not being absorbed and we don't want to give an injection or make an injection of this drug, or there could be various reasons. So as you go forward, and this is the end of the phase 3, where all of the data gets submitted to the FDA, you can see that all of these compounds that were being studied have been dropped, and finally, one drug could make it. What's happening here? What this is saying is there's a lot of attrition and there's a lot of time. Look at the number of years it takes. And can you imagine the cost? So there's a lot of expense that goes into this drug development before a drug gets onto the market. So, just stepping back, drug development is very complex. You already know that. There's so many components of it. And what I've just described is really very high level. And just to summarize, you go through compound selection. The chemist does that. There's compound selection. There's a lot of lab work that goes. And then there's the animal testing. So there's compound selection, lab work, animal testing, and then you do the human testing. You do small studies. Remember, phase 1 was smaller before you do large ones. Phase 3 are large studies, full blown studies. So, animals, then humans, small studies before you do large studies, short studies before you do long ones. So, remember there was one slide that showed acute, subchronic, chronic. So, you do short studies, even in humans. You can give single dose first, or you can give a few doses before you go into long chronic dosing into patients. You start with low dose before you go high dose. I mean, you would think these are all very intuitive, but this is the logic that is followed. A lot of caution and public safety and patient safety is the thinking that goes into drug development. Single dose before you start giving multiple doses. So, all of this data in the drug development progression, each step builds on the preceding experience. So each step of the way, there are lessons learned that are utilized and applied to the next step and the next step and the next step. And all of this data is gathered. And as you can imagine, it takes a long time. New treatments and cures take a long time for the drugs to be developed. Um, and and this is all the R&D, research and development process, and you don't even know 100% if that drug will be approved, because that assessment has to be made by the FDA based on the risk benefit. Do the benefits of the drug outweigh the risk of the disease and the risk of taking the drug? So at this point, the pharmaceutical companies don't even know. They've already spent so much time, they've already spent so much money, and along the way, there's a smaller and smaller and smaller percentage of the drugs that are moving forward. Even when you get into that clinical phase, the yellow phase, if you remember, when it go from the preclinical, all of the non-human, pre-human studies into the human studies, only 12% of the drugs that are into that stage for a drug to be approved. So you can imagine the relative magnitude, the expense, the time, all of that is basically is playing into the decision making as drugs are developed. And going back to the key slide that I'd shown earlier, now that I've gone through all of the drug development process at a high level, drug development is a complex process. It's a lengthy process. It's a multidisciplinary process. And it includes a collection of a lot of studies, a lot of information. And companies may find something in, let's say, animal studies or early human studies that's a surprise, and they have to go back and investigate that more, because they have to share that with the FDA in order for FDA to make a decision, a balanced decision of benefit and risk. So animal human safety studies, human efficacy studies, you can't just have a safe drug without its efficacy. You have to have safe and effective drugs. You have to have product quality. You can't have a drug that's developed that's very safe and very effective, but it doesn't have any shelf life, right? It can just sit on the shelf and it can just disintegrate, or it can just, there's no stability of that drug. So you have to make sure that it can be manufactured in large scale, and it could be manufactured and formulated into a dosage form, such as tablet or a capsule or a solution or a suspension or whatever is needed. Um, the product quality, the quality of the drug has to be maintained. All of these things are collected, all of this data are collected by pharmaceutical companies, and the evidence, all of that evidence is all collected and is sent to the FDA, Center for Drug Evaluation and Research, to review. It's called the New Drug Application, NDA or BLA, and the final decision on approval or non approval. And I've discussed this before, but it kind of gels in your head at this point, hopefully. It's based on the composite of all of the submitted data. And the ultimate approval decision is really based on whether the drug's benefits outweigh its risks for that intended population. So that's important, the intended population. So the bottom line is every decision that's made at the FDA is based on the benefit and the risk ratio for that intended population. And interestingly, if I pause here and just show you this, isn't this our life's decisions basically? Every decision that we make in our life daily. Do I get up at 8 o'clock? Do I get up at 6 o'clock? I have my exams at 8 o'clock in the morning. I have to set my alarm, so I have to get up. And if I ignore my alarm and don't get up, I miss my test. What is the risk benefit? What did I how did I make that risk benefit decision? And we do this every step of the way in our lives, risk benefit decision. And that's basically the principle on which FDA, CDER, and other centers they work. The risk benefit is really the core principle on which FDA works. And this is the end of my talk, and I would like to show you this slide here. It's basically training opportunities at the FDA. It's very fine here, but I believe the slides will be shared with you. To take a chance, take a look, uh, go through these if you're interested. There's a lot going on at the FDA. FDA is a huge agency. 17, 18,000 people, maybe more. 17,500 as of 2018 when I last saw the number. And about a third, maybe a little less, could be in CDER. And as I mentioned earlier, there are many centers. We are looking at a very broad range of things. We have a very broad mission. We have a lot of innovative research that's going on as well in unmet need areas. We are also responsible for um, ec- you know facilitating, supporting innovation in unmet diseases. That's part of our mission statement. And there's lots of training opportunities, summer training or ORISE fellows or various types of activities. And there are opportunities for students. When you get a chance, take a look at this. And thank you for your attention. Stay safe. And I'm glad I had the opportunity to talk to you.
Thank you, Dr. Ameeta Parekh. That was a lot of information and especially pertinent giving the rush for a vaccine for COVID these days. Now we have a better understanding of the process that it goes through. I have a quick question. You're very involved in policy, which is very fascinating to a lot of people, but how did you start? Did you start in research or did you jump right into a policy job?
Um, Very interesting question. So I got my PhD way back when, and a lot of my research was in animals. This may not be with everyone, but I hated research work in animals. At that time, after I finished my PhD,
Actually,
uh,
can I interrupt you for a second? Because one of our students posted a question that will fit right into what you're saying. How did you
how
ensure that testing on animals is as safe and painless for the animal as possible?
Yeah, when I did my PhD work, what a lovely question. I just thought I don't want to work with animals. And I decided I'm not going to get into animal research. I'm going to get into regulatory review of the applications, the research that already gets conducted by pharmaceutical companies. And I got fortunately, into the FDA, and I've been with the FDA for 34 years, and I have loved it. But let me answer Laura Bird's question here. How do you ensure that testing on animals is as safe and painless for animals as possible? Thanks. There are groups at the FDA who are actively involved and have the oversight. There are groups independent of the FDA who are keeping an eye on this. Eventually, we hope at some point that we can do without animal testing. Uh, we have initiatives where we are minimizing animal tests. There's something I mentioned at the end. I mentioned we are encouraging innovation in product development. One of the innovation is model development in lieu of, instead of certain types of studies. And we hope at some point that we get to a stage where we have enough in vitro tests where we don't have to do animal studies at this point. There are groups that are ensuring uh, minimal animal testing. Yet there are certain things that we are doing to get drugs approved. Yet we are pushing for research to, do more animal in vitro tests or developing in vitro models that can help us answer several safety questions.
That's great. Well, I think we are running out of time, but I know a lot of our viewers have more questions, so we'll post those and get them to you, and maybe you can answer them on your own, and we'll get back to
Absolutely.
them.
Absolutely, Mary Ellen. That's one thing I would like to say. Please share your questions with me. Go to our website. We are on the news a lot, but fortunately, a lot of our information, we are trying to be a very transparent agency. We have a lot of information on our website, and it's an education in and of itself. Please go to our website and try and understand what's going on. And on a day to day basis, we are on the news. So please go to our website and see what we do, how we do things, and why we do things. So it's
Yeah.
been a pleasure talking to you. And
Oh,
thank you
thank you
for
so much for your
inviting me.
time.
Thank you
Yes,
for
thank you.
inviting me.
All right. Take care and have a good afternoon.
Thank you. And thanks for the questions. Send me more questions and I will
Okay.
address them. Thank you.
All right. Take
Bye
care.
bye. Thanks.
Bye.
Bye bye.